Nobody told you that silence has a temperature. Not metaphorically, literally. When you go 24 hours without speaking to another person, your peripheral skin temperature drops measurably. Your hands get colder. If you put a thermal imaging camera on someone 12 hours into complete social silence, you would see it clearly. The blood has pulled inward. The peripheral vasculature has constricted, and the body has read the social silence as a threat signal and responded the way it responds to all threats. It protected the core. That's the first thing your body does. And most people have absolutely no idea. Let's start with the voice itself because it's the most obvious thing and also the most overlooked. Your larynx, the structure in your throat that houses the vocal folds, is a mechanical system that requires regular use to stay calibrated. And they're folds, not cords. Vocal cords is a term most of us grew up with, but it's anatomically wrong. They're two bands of mucous membrane stretched horizontally across the larynx. And when you speak, air from your lungs forces between them, causing vibration somewhere between 85 and 255 times per second, depending on your pitch and the person. That vibration does more than make sound. It maintains the elasticity of the tissue. It keeps the mucosal surface moist and functional. It keeps the entire mechanical system in working order. Stop for 24 hours and the changes begin within 6 to 8 hours. The mucous membrane starts to dry, not catastrophically, but the thin protective film that coats the folds during vibration, that film thins. And the mucociliary clearance mechanism, which normally sweeps debris and pathogens out of the laryngeal area during regular speaking, that slows down too. Because the mechanical action of speech is part of what keeps it running at full capacity. You stop providing the input and the system starts losing calibration. A 2019 study in the Journal of Voice followed trained singers through 4 hours of complete vocal rest and found measurable changes in the viscoelastic properties of the vocal fold mucosa. The folds became stiffer 4 hours. You are going to 24. But here is what makes the vocal fold story actually interesting and why it connects to something far larger than a dry throat. The act of speaking activates the vagus nerve, and the vagus nerve is one of the most consequential structures in the entire human body. The vagus, from the Latin for wandering, which is exactly what it does, travels from your brainstem all the way through your neck, your chest, your abdomen. It touches your heart, your lungs, your stomach, your intestines, your liver, your spleen. It is the longest cranial nerve in the body. And it has a branch called the recurrent laryngeal nerve that loops down into the chest and back up to your larynx. So when your vocal folds vibrate, when the muscles of speech engage, when air moves through that system, you are stimulating the vagus. Speech is in part a vagal activation event. Every conversation you have is quietly toning a nerve that governs your heart rate, your digestion, your immune signaling, and your inflammatory response. When you stop speaking entirely for 24 hours, you remove one of the most consistent daily activators of vagal tone. Not the only one. Deep breathing activates it. Cold water on the face activates it. Humming activates it. But speech is the one most of us do continuously from the moment we wake up. The contribution of speaking to overall vagal activity across a normal day is not trivial. A study published in Frontiers in Psychology in 2020 using wearable heart rate variability monitors on participants before and during a 10-day silent retreat found something specific and important. In the first 24 to 36 hours of imposed silence, heart rate variability dropped before it rose. That initial dip matters because it tells you something about how the body adjusts to losing its regular vocal input. The vagus is recalibrating. It's losing one of its consistent activators and hasn't yet compensated through other means. Heart rate variability, HRV, is one of the most reliable, non-invasive measures of autonomic nervous system health we have. High HRV means the nervous system is flexible, responsive, and adaptable. Low HRV means it's stuck, stressed, and rigid. Higher resting HRV is consistently associated with lower cardiovascular risk, better immune function, and more resilient stress responses. And on day one of silence, your HRV temporarily drops. Now let's talk about cortisol, because this is where most people get the story wrong. Cortisol has a reputation as the stress hormone, which is technically accurate but misleading. Cortisol has a daily rhythm. It peaks in the morning, roughly 30 to 45 minutes after waking, in what researchers call the cortisol awakening response. That peak is not a sign of stress. It's important. It primes the immune system, gets you alert, and prepares your body for the demands of social and physical engagement that the day holds. And a meaningful part of what keeps that rhythm healthy is exactly that: social engagement, conversation, the back and forth of being heard and of hearing someone else. A research group at Carnegie Mellon, published in the Journal of Psychosomatic Research, tracked the relationship between verbal social contact during the day and the cortisol awakening response the following morning in healthy adults. People with more verbal social contact showed more robust cortisol awakening responses the next day. People who had spent the previous day in low verbal or low contact conditions showed a flatter morning cortisol peak. The biological kickstart the body normally provides at dawn was blunted, quieter than it should have been. 24 hours of silence may flatten your cortisol awakening response the next morning. You wake up and the system doesn't fire the way it's supposed to. Whether that's harmful or restorative depends entirely on context. And this is the part of the silence conversation that almost nobody handles honestly. There are two physiologically distinct states that look identical from the outside. Both involve zero verbal communication for 24 hours. One is chosen silence, a retreat, a deliberate day alone, a spiritual practice. The other is isolation, the kind that happens when you are depressed, estranged, grieving, or simply forgotten. Same observable behavior, completely opposite internal biology. When silence is chosen and felt as safe, the nervous system reads it correctly. The anterior cingulate cortex, the region of the brain that continuously monitors for social threat, quiets down. The vagus activates more deeply. Cortisol stabilizes rather than dysregulates. The body actually rests. When silence is imposed or unwanted, when there is nobody to call, when you are at the edge of other people's lives and nobody has come for you, the anterior cingulate cortex does not quiet. It stays on. It reads the absence as danger. And that distinction matters enormously for what happens in the blood. This is where the work of Steve Cole at UCLA becomes impossible to ignore. Cole is a genomics researcher, and his work on what he calls the social genomic regulation of gene expression is among the most important and least discussed research in modern medicine. What his lab has shown across multiple studies going back to around 2007 and continuing through the following decade is that loneliness, specifically the subjective experience of social disconnection, alters the way specific genes are expressed in immune cells. Not hormone levels, not behavior, the actual transcription of genes. In people reporting high loneliness, immune cells showed upregulation of genes involved in inflammation and innate immune responses. The blunt, fire everything response your body uses when it thinks a physical attack is coming. Simultaneously, those same cells showed downregulation of genes involved in antiviral and antibody-mediated responses, the targeted intelligent part of the immune system that fights specific pathogens. That trade-off is evolutionarily logical. If you're isolated and abandoned, your ancestors in that situation faced elevated risk of physical injury from predators or conflict. The body prioritizes wound defense. It pools resources from targeted immunity and redirects them toward blunt inflammation. The problem is that in modern life, this ancient immune recalibration is not protective. Chronically elevated inflammation is linked to cardiovascular disease, type 2 diabetes, neurodegeneration, and certain cancers. And a chronically suppressed antiviral response is obviously the opposite of what you want in any world that contains respiratory viruses. Those signals begin shifting within 24 hours. Cole's work primarily documents patterns in people experiencing chronic loneliness across months and years, not a single quiet day. But in a 2015 paper in the Proceedings of the National Academy of Sciences, his group found measurable changes in leukocyte gene expression profiles after relatively short periods of social disruption. The trajectory starts early. The direction is established quickly. Let's come back to something purely physical for a moment. The gut. Your gut contains somewhere around 100 trillion microbial cells. More microbial DNA in your colon than human DNA in your entire body, which is a fact I find disorienting every time I think about it carefully. That community of organisms, the microbiome, is sensitive to an extraordinary range of signals from the rest of the body, including the neurochemicals and hormones that shift in response to social contact or the absence of it. The gut-brain axis is bidirectional. The vagus nerve is one of its primary communication pathways. So when vagal tone shifts, as it does when you stop speaking for a day, the signals running down to the gut shift too. The enteric nervous system, which is the network of roughly 500 million neurons embedded in the lining of your gut and sometimes called the second brain, is receiving different information and it responds. A 2020 study from the German Center for Neurodegenerative Diseases found that socially isolated subjects developed measurable changes in gut microbiome composition within 72 hours, reduced microbial diversity, lower populations of certain lactobacillus species, elevated levels of inflammatory bacterial strains, and those microbiome changes corresponded with behavioral markers of anxiety. The subjects in that particular study were not human, and I want to be precise about that rather than pretend otherwise. But the mechanisms are conserved enough across mammalian species that the direction of the finding applies, even if the exact magnitude differs. Your gut notices when the social input stops. Now, the brain, specifically the default mode network, when you are not focused on any specific external task, a network of brain regions activates the medial prefrontal cortex, the posterior cingulate, the precuneus, parts of the temporal lobe. This network, the DMN, is sometimes called the resting brain, which is completely inaccurate. It's not resting. It's doing the most distinctively human work the brain does. It consolidates autobiographical memory, processes emotionally charged events, simulates future scenarios, builds the narrative of self. The DMN is where your brain makes meaning out of experience rather than just recording it. Most of the day, conversation suppresses it, not entirely but meaningfully. Social interaction demands external focus. Tracking someone else's words, reading their intent, processing tone and subtext. Those networks are in partial competition with the DMN. So constant speaking inadvertently suppresses the brain's capacity for deep, quiet, internal processing. A day without conversation frees the DMN to run long. And that is double-edged. A DMN running on chosen solitude and safety can produce something close to insight. Connections between ideas and experiences that were too fragmented to form in the noise of a social day. Emotional processing of material that had been sitting unresolved for weeks. The clarity that sometimes comes after a long solitary walk, where you suddenly understand something you'd been confused about. That's the DMN completing thoughts the day had kept interrupting. But a DMN running on loneliness and anxiety doesn't produce insight. It produces rumination. It cycles through the same emotional material without moving. It revisits without resolving. And rumination is one of the clearest predictors we have of depression onset in otherwise healthy adults. A 2021 meta-analysis from Oxford across 43 studies and over 9,000 participants found that trait rumination was the single strongest individual predictor of depressive episodes over a 1-year follow-up period. Stronger than sleep disruption, stronger than stressful life events, stronger than almost any other single variable. The same silence that nourishes one person's DMN feeds another person's rumination loop. And which one you get has almost everything to do with why you're silent. Oxytocin gets described as the love hormone, the cuddle chemical. And those descriptions are accurate but dramatically undersell what this molecule actually does in the body. Oxytocin is produced in the hypothalamus, released from the posterior pituitary, and it touches an enormous range of physiology. Social bonding, yes, but also pain perception, inflammatory regulation, wound healing speed, cortisol management, cardiovascular stress response, neuroplasticity. The triggers for oxytocin release include physical touch, eye contact, shared laughter, and voice. Specifically, the human voice in social context. The back and forth of conversation, of being heard, of understanding, and being understood. Go 24 hours without any of that, and oxytocin falls. The magnitude of the drop varies by personality type and baseline social activity. Introverts and extroverts have genuinely different oxytocin response profiles, documented independently in at least 3 separate labs. But the directional change is consistent across people. With reduced oxytocin, pain sensitivity increases subtly. Inflammatory tone increases. The cardiovascular stress buffer gets a little thinner. The body is slightly more exposed than it was yesterday. And then there's serotonin. The serotonin synthesized in the Raphe nuclei of the brainstem and distributed across cortical regions governing mood, impulse control, and complex decision making. Serotonin is not independent of social behavior. Michael Raley's work at UCLA on vervet monkeys in the 1980s showed that dominant, socially connected animals had significantly higher whole blood serotonin than subordinate or isolated ones. When dominant animals were isolated, serotonin fell within days. When isolated animals were reintroduced to social groups, it rose again. The human serotonin system shows the same directional sensitivity to social context, though measuring it directly in people is harder. What matters practically is that serotonin heavily modulates the prefrontal cortex, the seat of planning, impulse control, emotional regulation, and the kind of flexible, considered decision-making that separates a thoughtful response from a reactive one. When serotonin tone in the prefrontal cortex drops, decisions under uncertainty become more risk-averse. Emotional flexibility decreases. The precision of behavioral control quietly degrades. After 24 hours of unwanted silence, you may make slightly worse decisions the next day. Not dramatically, not enough to trace back to the silence, but the precision was reduced. Blood pressure, speaking raises it slightly every time. the muscular effort, the respiratory adjustment, the social engagement. Blood pressure during speech is consistently a few millimeters of mercury above resting baseline. So silence should lower it, and sometimes it does. A 2006 study in the journal Heart found that silent rest in a safe setting produced genuine parasympathetic activation and blood pressure reduction. The silence condition outperformed music and breathing exercises for immediate cardiovascular quieting. But that was laboratory silence. Safe silence, observed silence. In the silence of unwanted isolation, blood pressure behaves differently. The brain reads social absence as threat. Threat activates the sympathetic nervous system. A 2018 study from King's College London, published in JAMA Internal Medicine, followed over 4,000 adults and found that those reporting high loneliness had systolic blood pressure readings 4 to 6 millimeters of mercury higher than socially connected adults. After controlling for age, BMI, physical activity, and every obvious confounding variable. 4 to 6 millimeters, that sounds modest, but sustained over years in population level cardiovascular data, that elevation translates to meaningfully increased stroke and heart attack risk. One day of silence points in that direction. John Cacioppo spent his career at the University of Chicago building the epidemiological and biological case that loneliness, not measured as a head count, but as the subjective sense of disconnection, is a stronger predictor of early death than obesity, physical inactivity, or moderate to heavy smoking. The number that gets quoted is roughly a 26% increase in premature mortality risk. But that number strips away the mechanism, and the mechanism is what matters. The pathway is biological: inflammation, immune dysregulation, disrupted HPA axis function, cardiovascular stress, degraded sleep architecture. All of the things I've been describing compounding every day across years. One day is not loneliness, but one day is the first data point on a spectrum. Sleep, because if your 24 hours of silence includes a night, which it does, the architecture of that night is altered by what the day contained. Sleep cycles through distinct stages: light sleep, deep slow wave sleep, REM. Each full cycle takes roughly 90 minutes. Slow wave sleep is when physical restoration happens. Growth hormone is released, cellular repair accelerates, and the lymphatic system flushes metabolic waste from the brain, including amyloid beta proteins, the ones that accumulate in Alzheimer's disease. REM sleep is when emotional memory consolidation happens, where the brain processes charged experiences from the day and integrates them into long-term memory in a chemically de-aroused state. When cortisol is elevated from the stress of unwanted social silence, it suppresses slow-wave sleep. The body stays in lighter stages longer. The restorative architecture degrades. When oxytocin is lower, the nervous system enters sleep less buffered, more easily disrupted, and harder to push into the depths where the most critical repair work happens. The lymphatic system runs less efficiently in lighter sleep, the cellular repair is incomplete. And the social engagement of a normal day, the verbal exchange, the processing of other people's words and intentions and emotions, loads the hippocampus with material that drives sleep pressure at night. Conversation creates memory traces. Memory traces generate sleep need. A day with no social input and no verbal processing generates a different sleep drive entirely. The hippocampal load is lighter. Some people sleep less deeply after a solitary day. Some sleep more easily because the social noise isn't buzzing through the night. The research genuinely varies, but the sleep you get after 24 hours of complete silence is almost certainly not structurally identical to the sleep you get after a day of normal human contact. The architecture shifts, and which direction it shifts depends, again, on the quality of the silence rather than its mere existence. Let me come back to the larynx one final time. The vocal folds form in a fetus at around 6 weeks of gestation. Before there are lungs to push air through them, before any neurological wiring for speech is in place. They form before any of that. And when a newborn takes its first breath, the very first thing the body does with an airway is make sound. Not intentionally, reflexively, but immediately. The tissue exists before the function. And then, for the rest of a human life, requires the function to maintain itself. In patients who go beyond 5 to 7 days without any phonation at all, due to surgery, nodules, or illness, the vocal fold mucosa undergoes structural changes, not just surface dryness. The superficial layer of the lamina propria, which is the layer just beneath the epithelium and is critical for the smooth, elastic vibration that produces a normal voice, loses fluid content and elastic fiber organization. These changes are reversible, but they don't reverse on their own. The tissue needs the function to restore itself. The structure depends on the behavior. 24 hours will not structurally alter your vocal folds, but it points in a direction, and this is the actual story of a day without speaking. Not catastrophe. No single day of silence causes catastrophic harm. The body is resilient and redundant, and it tolerates a great deal. But everything I've described is directional. The HRV dips. The cortisol awakening response flattens. Immune gene expression moves toward inflammation. Oxytocin falls. The serotonin modulation of the prefrontal cortex thins. The gut microbiome receives a different signal. The DMN either nourishes or ruminate, depending on something that no blood test can measure. And the vocal folds begin the slow drift away from the elasticity that comes from being asked to do the thing they formed to do before you even had. Lungs. Cassiopo said something I've thought about regularly since I first came across it. He said, loneliness is not the absence of people. It is the absence of a sense of being known. You can be in a room full of people and feel it completely. You can be genuinely, physically alone and not feel it at all. Your body doesn't respond to a head count. It responds to a signal, a pattern of inputs arriving through 100 subtle biological channels that says, collectively, I exist to someone. I am registered. Someone's nervous system has made room for mine. 24 hours is long enough for the body to notice when that signal goes quiet. The question that no study has fully answered is, how many people going a full day without speaking to anyone are actually choosing it? And how many of them are simply waiting for someone to call, waiting for someone to notice, waiting in a silence that is completely indistinguishable from chosen solitude if you observe it from the outside? From the outside, they look exactly the same. From inside, the blood, they never did.